For thousands of years, extinction meant gone forever, a dead end. But the biotech industry has set a very different, highly public timeline. They are currently promising to produce a living, breathing woolly mammoth calf by 2028. To see how serious they are, you don't have to wait until 2028. A Dallas company called Colossal Biosciences is already raising 3 living dire wolf pups. 2 males named Romulus and Remus and a female named Kessie are growing up right now on a secret fenced preserve. But the biological reality of how those pups got there is hidden in the fine print. Scientists cannot extract a complete strand of ancient DNA to resurrect a dead species because fossilized DNA fragments over time. Instead, they take the intact DNA of a living relative, like a gray wolf, and rewrite it using the ancient fragments as a guide. The sheer scale of that genetic blueprint is massive. A dire wolf differs from a gray wolf by millions of bits of DNA. To create Romulus, Remus, and Kessie, Colossal actually altered only about 14 genes out of roughly 19,000. The animals walking around that private preserve are not resurrected from the Ice Age. Even the company's chief scientist calls them gray wolves with edits. They are modern animals wearing a very precise genetic costume. A 100% genetic resurrection is technically possible, but the baseline for how that actually plays out in the real world was set 20 years ago. In 2000, the last Pyrenean ibex, a wild mountain goat from Spain, died. But before she did, scientists froze a scrap of her skin. This allowed for true cloning. Researchers took an egg from an ordinary farm goat, scooped out its DNA, and slipped the dead ibex's preserved DNA inside. A jolt of electricity started the growth process, and the egg was placed into a living surrogate oat. In July 2003, it worked. A baby ibex was born alive. But one of her lungs had formed wrong, making it impossible for her to process oxygen. She suffocated and died in the researchers' hands, just minutes after taking her first breath. She was a perfect copy of a single dead female. Even if she had survived, she had no mate and no herd to join. Because of that biological dead end, the Pyrenean ibex became the only animal in history to go extinct twice. Look at this 110-year-old preserved specimen. Because it was kept in a jar of liquid to prevent rotting, scientists at the University of Melbourne were able to extract an almost complete gene code for the thylacine, the extinct Tasmanian tiger. But having the recipe isn't enough. You need a biological base. Scientists are rewriting genes of the thylacine's closest living relative. It is a tiny marsupial called a dunnart, which looks like a small mouse. The researchers are making over 300 genetic changes to dunnart cells to try to build an animal that eventually looks and acts like the actual thylacine, a striped, dog-shaped meat eater. This same surrogate logic is being applied across the world. To rebuild the blue buck, a South African antelope shot out of existence around 1800, scientists take faint traces of DNA from a 200-year-old Swedish museum specimen and narrow down differences against cells of this living rowan antelope. They are hunting for the 20,000 specific genetic differences that matter. But no matter how perfectly they edit those traits into rowan cells, the calf that grows from them will not be a blue buck. It will be a completely new biological hybrid. Rebuilding lost traits is entirely different from physically birthing the animal, especially when dealing with eggs. To bring back the dodo, scientists are isolating traits from its closest living relative, the Nicobar pigeon. You cannot clone a bird directly. The biological workaround is extremely complex. The team is growing special pigeon cells that carry dodo traits, and then placing those modified cells into ordinary chickens. The hope is that eventually those chickens will lay eggs that hatch into something resembling a dodo. The moa, a flightless New Zealand bird wiped out 600 years ago, presents an even steeper physical barrier. A grown moa was taller than a human, and its egg was roughly 80 times larger than a standard chicken egg. No living bird is physically capable of incubating it. To bypass the size limit, scientists built a massive man-made eggshell from scratch, designed to grow a chick entirely without a mother. In May 2026, they successfully hatched healthy, ordinary chicks inside those artificial eggs to test the hardware. Mapping a dead animal's genome yields nothing if science cannot replicate the complex physical mechanics required to gestate it. Without a mother or a functioning artificial womb, those highly engineered cells stay trapped inside laboratory glass. All of this brings us back to that 2028 deadline for a woolly mammoth. The biological foundation for that promise is the Asian elephant, the mammoth's closest living relative. By altering the elephant's genes, the researchers are pacing in specific traits: thick fur, smaller ears to prevent frostbite, and a heavy layer of fat. A living elephant mother will then carry that modified embryo until a calf is born. When that calf finally takes its first breath, it will have no mammoth mother to teach it, and no ancient herd to follow. Is it actually a mammoth, or is it just an elephant wearing a mammoth's coat? Even if a perfect physical match is born, the world it belonged to is gone. The island of Mauritius, the only place the dodo ever lived, is now heavily developed. The ecosystem it waddled across 300 years ago simply does not exist anymore. These projects prove that science can manipulate living cells into incredible shapes. We just have to decide if it makes sense to spend millions of dollars building living lookalikes of animals we killed centuries ago, or if we should use those exact same resources to save the authentic animals still breathing right now.